Hello everyone, my name is Edmund and I'm a bioinformatics engineer on the scientific development team at Seqera. In this role I work closely with customers, helping them get the most out of their software and their workflows. One of the things that we often see is that as pipelines evolve, growing in scope and complexity and the accumulating challenges along the way become a barrier for our customers. So in this talk, I'll walk through some of the common pain points and how the Seqera ecosystem is built to reduce friction at every stage so that your focus can stay on where it belongs, on the science. So we're going to talk a lot about Nextflow today, so let me start off with a brief introduction. Nextflow is a programming language designed for building bioinformatics pipelines. It was created at the CRG in Barcelona in 2013, and since then it's gained widespread adoption for its simplicity and its flexibility and its ability to scale across a range of computing environments. Over the past decade, Nextflow has become the industry standard in both academia and industry. So in Nextflow, you have steps in your analysis, and each step is defined as a process, and the data is passed between them via channels. You can think of this kind of like a metro map. The processes are stations along the subway, and the channels are the tracks, and the data is flowing through as the train is moving along the lines. One of Nextflow's key strengths is how it abstracts away the execution logic, and it automatically determines the correct order of operations and runs tasks in parallel where possible. Its configuration system allows you to run the same pipeline across a range of environments, whether that is your laptop, a cluster, or the cloud. This intuitive structure is a big part of why Nextflow has become so popular among bioinformaticians. Seqera is the company founded by the team behind Nextflow. Our customers, our founders, Evan and Paolo, created Nextflow, and we continue to develop it as an open source project. While Nextflow has been transformative for individual bioinformaticians, but Seqera was created to meet the broader needs of these organizations with centralization, standardization, and management. So with Seqera, Nextflow evolves from a powerful workflow engine into a full platform for collaborative science. So I've introduced Nextflow, Seqera, and now let's talk about the challenges that bioinformaticians face. One thing that I often see when working with customers is the transition from small Nextflow to big Nextflow. The core evolution from a simple single script to a fully tested portable production grade pipeline. It starts with a single script and a concise workflow. Then the features are added iteratively. So as a pipeline uses more context and more purposes, it naturally grows, and as soon you have a fairly complex piece of software. But there are challenges at every stage. It might begin with a single step in the pipeline, but it quickly grows into a complicated mess that requires proper packaging, versioning, and deployment. So you go from running locally to HPC clusters to the cloud, and then tracking the when, where, and how of a pipeline as these runs becomes a full-time job, especially when the same pipeline is reused across multiple projects. And lastly, running the pipeline isn't enough. You need to interpret the results and make meaningful decisions. So these are roadblocks at each level of going from small to big Nextflow, and these are compounding problems that slow down your science. So let's talk about each of these 4 points in turn, starting with software. So if you want to build a complex piece of software, good developer tools are essential. So we've just rolled out a VS Code extension, and it's making developing, debugging, and expanding your pipelines so much easier and faster. So now you get a full IDE experience right in VS Code. And that means things like syntax highlighting, error checking, and even a graphical view of your workflow, which you can use to navigate your code. So all these features make it easier for you to develop and debug your pipelines. And so there's an added bonus. We're also hooking these tools into other parts of the Seqera ecosystem, like handling containers and even AI-powered suggestions. So it's all about helping you build pipelines quicker and more efficiently. And of course, it's 2025, so if you're still getting stuck, you can just ask the machine. So we've also developed Seqera AI, a language model optimized specifically for working with Nextflow and bioinformatics. It's designed to help with everything from identifying and fixing errors to converting existing pipelines into Nextflow. And also you can just use it to generate a brand new workflow from scratch. So importantly, it will not reinvent the wheel. It will use the best practices and resources from the Nextflow community, such as NF Core, where possible, to ensure you are using the best possible tools. So Seqera AI is directly available from our website. It's also built right into the Nextflow VS Code extension through GitHub Copilot, so you can get real-time help without ever leaving your coding environment. Next, containerization is also the right way to package pipeline software. It ensures stable environments and arguably the only way to do reproducible scientific analysis. But creating Docker and Singularity containers isn't the best use of a bioinformatician's time. They should be doing science, not wrangling containers. That's exactly why we created Wave, a container provisioning service that automatically builds and deploys containers based on your needs of your Nextflow pipeline. So just specify the software requirements for each process, and Wave takes care of the rest. So we've wrapped Wave also into a public service called Seqera Containers. Just visit the website, select the software packages you need via Conda or pip, and Wave will build a container, publish it to a public registry, and complete a security scan as well. So each of these tools works together to build a bigger and more help you build bigger and more complicated pipelines with confidence. But we're developing on our laptop locally, and what happens when we run out of storage and we need more horsepower? We need to increase our computing capacity. If we have access to an HPC, we could use that, or we could also deploy into the cloud, like AWS, Azure, or GCP. But these require considerable amounts of experience to set up, and if you're not a DevOps or sysadmin, you're a bioinformatician. So again, it's taking away time from your core job of computational science. Okay, so next, this is where Seqera Platform comes in. Seqera Platform is our primary interface for managing and running Nextflow at any scale. One of the cornerstone features of Seqera Platform is its ability to create and manage compute. You can easily spin up environments on cloud or on-premise directly from the interface without needing to dig through complex options. These pipelines can then be launched onto these compute environments straight from the platform with no need to supply extra configuration or credentials. Just select the environment and go. It's also purpose-built for bioinformatics workloads with features like automatic scaling, resource caps to control costs, and they also include support for spot machines, GPUs, and ARM, reflecting all the diverse hardware requirements of bioinformatics. But if that feels like too much work, we've recently introduced Seqera Compute, a fully managed compute service provided by us. And so when you choose this option, all you need to do is pick a geographic location and give it a name, and we'll handle everything else. So Seqera Compute brings everything together that we've learned from running production workloads across a range of environments and packages into a single ready-to-use solution. And so one of the features here that we'd also like to call out is that we're also adding in free compute for new signups on Seqera Compute as well, so you can try out Platform in an actual HPC environment, even if you don't have access to one. And so this integration requires minimal configuration changes to your Nextflow workflows into Seqera Platform, and then there's also no complex infrastructure setup needed, so it's automatically all handled through Seqera Platform. One of the features as well is that we have GPU enablement just by clicking a button to use the correct AMI, and that sets up all the right drivers and everything to use GPUs. And so Gary is going to tell you a little bit more about using GPU workflows in Seqera Platform.
Hey everyone, my name is Gary Burnett. I'm a tech marketing engineer here at NVIDIA, and my job is to help users get started with NVIDIA software, particularly on the genomics team. And so what I'm going to talk to you all today is with what NVIDIA is doing with GPUs in bioinformatics and how that's going to work with Nextflow. So I'll start off by going, why would you even want to use GPUs? Right? What would you use GPUs for in this context? And the answer is if you have a large amount of data that you want to process very quickly. And that's what this graph is kind of showing. And so in this solid gray line, we have the cost of sequencing over time. And as we've noticed, as the cost of sequencing has gone down, it's been easier and easier, so people are sequencing more and more data. And so now the amount of data shown in this green line, genomics data in public repositories, is exploding. And so the bottleneck in these workflows has now shifted from the actual sequencing to the data processing. And so now it's time for the tools that we use in these bioinformatics workflows to actually start keeping up pace with that. Hence what NVIDIA has been working on the last couple of years in genomics. What we've been doing the last few years is basically taking a lot of the popular bioinformatics tools that anyone would be familiar with and porting them over to the GPU. And so those tools that we have fall into these three buckets, represented by these three boxes here in green. And so these first two boxes cover alignment and variant calling tools, which are under this umbrella project called Parabricks. But to fill out the whole ecosystem, we also have tools for single cell and spatial further downstream. But for this talk, I'm going to focus mostly on Parabricks and the tools that we have there. So if you take a look at what's listed in these green boxes, you'll see aligners, right, such as Giraffe, BWMM, Minimap2, right? And similarly with variant calling, you'll hopefully be familiar with tools like Deep Variant and Deep Semantic and Mutex2. And so what the Parabricks team has done at NVIDIA is basically taken a lot of these popular tools that already exist in the ecosystem and moved them over to the GPU. And so now we have all of these tools and more available as drop-in replacements for the tools that someone might be using today. And so why would you use these Parabricks tools over maybe the ones that you're familiar with? And the answer would be because these are running on the GPU, what you get is an increase in speed, right? So these tools will give you the same results that you've gotten before, but much, much faster. And with the speed increase also comes a reduction in cost. And so to go a little bit more detail into what the actual numbers on that look like, I can go to. So to go into some more of these numbers, these are some benchmarks that I'm showing for a 30x whole genome sample, and we're going to compare running them on the CPU versus running them on the GPU. And the workflow that we're going to run is a germline analysis, and so both alignment and variant calling all the way from FASTQ to BAM. It takes about 16 hours to run this sample on the CPU, and that's represented here by this dark gray bar. Now, using the Parabricks replacements for these tools, running them on GPU, is what's shown in these green bars. And so this will take the runtime down from 16 hours on the CPU to about 10 minutes on the GPU, depending what GPU you're using. And like I said on the previous slide, the accuracy is exactly the same as you would expect on both of the machines. But the only difference is the runtime, and therefore the cost as well if you're running this, especially on the cloud. Being able to run 100 times faster can save you a lot of money in your compute costs. And so this is really what the goal is of running these tools on the GPU. And what I'll talk to you next about is some of the more specific updates in the latest version of the software that was just released at ASHG this year. This week at ASHG, Parabricks 4.6 was released and includes these four major updates across Deep Variant, Deep Semantic, Star, and Mutex. I'll touch lightly on the Deep Semantic and Mutex updates and then double click on Deep Variant and Star further on. So with Deep Semantic, now we've moved on to version 1.9 to support long read and whole exome sequencing. And with Mutex Color, we now support additional arguments for mitochondrial mode. And so really, as these tools are improving and as new versions are being released, we're just trying to add more functionality to the GPU runtimes. Now to double click on Deep Variant with Parabricks, Deep Variant has now been bumped to version 1.9, and the big announcement there is that this supports for pan-genome workflows. And so using pan-genome over Deep Variant, you get all the benefits you'd expect of the pan-genome, which is a reduction in errors of up to 25%, which is actually huge. And this is especially true in diverse populations when you get to the variant calling, just by representing these variants with the graph. So this is what you normally things that you would expect from Deep Variant, but specifically with Parabricks Deep Variant, you'll get speed ups as well. And so our GPU-accelerated Deep Variant is 14 times faster than the CPU version, which takes your runtime down from 9 hours to just under 40 minutes. And so you get all the benefits that you would normally get with pan-genome Deep Variant, but now you get them much, much faster. Similarly with Star and this new version of Parabricks, there have been speed improvements to Star, so now Star is 8 times as fast as its CPU counterpart, and it now supports this new quant mode for gene counts, which is going to be really critical for downstream analysis when you start to do QC and normalization. It's going to be really important to have those gene counts and to have them quickly. So that was my summary and overview of running these bioinformatics tools on the GPU. To bring this all back to Nextflow, all these tools that I've talked about today in their latest 4.6 version are available as Nextflow modules up on NF Core. And so I encourage you all to try them out and go and see how you can incorporate GPUs into your workflow today. And so with that, I'll pass it back to Edmund to talk about some of the GPU experiments he's done with Nextflow and NF Core.
Okay, thanks, Gary. So now with Seqera Platform, we can use the distributed compute environment and scale even bigger, including with GPUs. So the nice benefit of this is then we can start to use Fusion, which is our high-performance virtual file system designed for bioinformatics workflows. What Fusion does is it streams data directly from cloud storage during execution and eliminates the need to download or upload files, which is a major bottleneck when using GPU-enabled instances in the cloud. So this delivers two key benefits. Firstly, faster pipeline execution by removing the data transfer, and secondly, the freedom from managing intermediate storage. So your teams can focus on the analysis rather than the infrastructure. So what we did was we wanted to see what happens when you combine the two powerful technologies, Parabricks for GPU-accelerated genomics and Fusion for optimized data access. So we ran NF Core Seqera, which is a pipeline that anyone can pull off of the shelf, and it's a whole genome sequencing pipeline. And we ran it across a couple of different compute configurations. So first, we ran it with CPU only to get a baseline. It took nearly 20 hours for a complete run. And then next, we started to run it on an L4 GPU instance. And so we saw the runtime drop to about 2.5 hours, which is already a massive improvement. And then we also tested on an A10G GPU. And so that was under 2 hours end to end. So that's a 10x speed up compared to CPU. And so now faster doesn't always mean better, right? Depending on the cost with bioinformatics, I think bioinformaticians are very cost sensitive with the large compute that we're doing. This isn't, speed trading. So we did a cost analysis next. So we kind of wanted to go and rerun them again. So the CPU run was $40. And then the L4 instance was about $25. And then lastly, when we ran out the A10G, we got it down to about $14 a run. So we're not just running faster, we're also cutting costs by 65% when we're getting a 10x speed up. And this is also while maintaining the exact same analytical quality. So the same results, fraction of the time and the cost, and this is due to Fusion being able to load these files directly from the S3 file system directly onto the disk and stream it in versus having to stage everything in and stage everything out at the end. And so another thing that Fusion and Seqera Platform allow you to do is to use spot instances. And so without really adding much complexity to this, we were able to rerun this with spot instances. So I've got the $40 for the CPU and the $14 for the G5 instances on there, and these allow us to have like some fault tolerance as well whenever the jobs are interrupted, and then we can, with Fusion snapshots, we can start to re-enable the job part of the way through. And so by leveraging spot pricing with CPU, it drops to about $13, which is actually pretty competitive with the on-demand GPU pricing, and kind of negating our effects of the gains from the GPU usage. However, then when we start to use spot with the GPU instances, it also drops to around $13, it drops from $14 to $6. And so that's actually really competitive comparatively. So we've gone from $40 all the way down to $6 to do a whole genome sequencing run with the same results on that. And so not only is that like a increase in productivity, and it's also an increase in the compute budgets, and it allows your dollar to go a lot further just by using some of these technologies. So now with Seqera Platform, we can see that we can use a distributed compute environment and scale bigger than ever. Your pipeline is now in active use though in this scenario. So that means you need to keep a complete, accurate log of how, when, and where each analysis was run. So let's move on to projects. So keeping track of all these pipeline runs can be a real challenge. You could try to document everything manually, but there's no guarantee it'll be accurate or complete. So just to make sure, you want to capture all the parameters, settings, but what about the deeper details, like pipeline configuration or the internals of an individual tasks? So with Seqera Platform, every pipeline run is monitored end to end. No more digging through emails or Slack threads to find the right configure output. It's just there. So it all lives together in one place, and you can follow the execution live. And then after the run, you get access to detailed metrics and logs all the way down to the task level. So these errors are also reported in plain text right inside the platform. Failed tasks are clearly highlighted on the run page, so your attention goes exactly to where it's needed, and you're not sifting through logs in your various cloud platforms. So you can also dive deeper without switching tools. You can download these logs directly from the platform or explore the task directories using the built-in data browser. So debugging becomes faster, more focused, and far less painful, and frees up the bioinformatician's time to focus on improving pipelines rather than debugging them. So this gives you a complete and accurate view of how your pipeline ran automatically with zero effort from you. When someone asks you how you did a specific analysis, you can point them to the pipeline run on the platform. And so pipelines are all grouped under workspaces, which grant you specific role-based access to only the relevant details at the right time. You can invite other collaborators, whether internal or external, and give each one the access they need. You're working from the same source of truth, all together. So these workspaces bring clarity and consistency to the collaborative science experience. So now we've seen how Seqera Platform provides a unified view of your pipelines. Analysis doesn't just stop with pipelines, though. Results need to be interpreted and presented clearly to make a conclusion. And so these bioinformatics results are often messy and nuanced, and that makes them difficult to communicate, especially to non-bioinformaticians. So figures are usually the best way of conveying a message, but generating one usually involves parsing raw outputs, standardizing formats, and building plots. And so that's where MultiQC comes in. It's a tool maintained by Seqera and was built exactly to solve this problem. It aggregates and standardizes results from your pipeline into a single, easy-to-read report. With built-in support for 165 bioinformatics tools, you don't need to write a custom parser to get a clear, representative view of your data. So if a tool you use isn't supported out of the box, MultiQC can be extended using its Python-based module system. If you need more flexibility, you can also import MultiQC directly into your own Python scripts to generate a fully customized report. MultiQC makes it easy for your team to interpret outcomes and make informed decisions. And so MultiQC just recently turned 10, and it's freely available as well for the community. And so a pipeline is ultimately a transformative engine, but without analysis and interpretation, it's incomplete. Too often, analysis happens downstream. Users download pipeline outputs to local machines and work in isolated environments, and this breaks reproducibility and increases the risk of errors. So Studios is our version of an interactive analysis environment. So there's three core advantages: runs inside of your compute environment that you're using for your Nextflow pipelines already, and it launches directly into this configured infrastructure, allowing you not to have to create a separate environment for analysis and pipelines. It's all backed by the same thing. It's also backed by Fusion for instant data access, so this allows you to stream data directly from your cloud storage and no need to copy data from one source to the other onto large local disks or copying all of these files. It also supports the tools that you need, such as Jupyter, R, VS Code, and even full virtual desktops. So you can use things like IGV or Cell by Gene, as you can see below in the example. And so all of these can be customized using Conda, and finally you can bring your own custom containers as well to the Studio experience. So Studios is an ideal shared analysis space for computational biology teams, and it empowers you to turn data into decisions. So by providing the tools for analysis, we've turned raw pipeline results into informed, actionable decisions. And so this is the core mission of Seqera: to unblock bioinformaticians at every step by removing technical friction. You shouldn't be spending your time managing infrastructure, juggling software environments, or tracking down results. Your software or your science should not be slowed down by the work around the work. And so here's a list of all of the products that we've mentioned in this talk. If you want to discuss any of these further, please reach out to us or reach out to Gary as well if you'd like to talk more about Parabricks and using GPUs. We also have the Nextflow Summit coming up on October 23rd and 24th that is virtual. Thank you all for listening.